There are countless streams of data always pouring in to the ship at any given moment. These streams of data are telling us about the chemistry of the water, the physics, the temperature, the currents, and stuff above the water too, like weather and winds. The data sets that Sekuliak is able to collect on our behalf while we're out here really help us put our work in a strong oceanographic context. We have a couple of ways to analyze particles, and one of them is with this big instrument package here, the CTD rosette. Strapped to the bottom of it, we've got a few different sampling devices, one of which is an underwater microscope, and it takes pictures of particles through the water starting out about the size of a human hair and going up to a few centimeters. And with that we can analyze the sizes, the types, and abundances of particles all throughout the ocean. We can do that every time we put this in to take our standard measurements of physics and chemistry with the CTD rosette.
In my world, the big giant organisms can be about 100 to 200 microns. Now for a sense of scale, the width of a human hair is about 100 microns. A lot of the organisms that I work with are 1 to 5 microns, and there are a lot of them, even in a drop of seawater. What has been developed over the last 10 to 20 years is something called flow cytometry. In flow cytometry, you take a water sample or any sort of liquid sample and you pass it by a laser. The laser is very focused and very bright and tuned to a specific wavelength. So we use something called the flow cam, which works on the same general principles as a flow cytometer. So you have a laser to look at particles passing by, but in this case, anytime the instrument detects a particle, it actually snaps a picture. And so that way we're able to look at not only how much fluorescence and chlorophyll there is there, but we can actually start to identify. So I have some examples up on the screen of different types of phytoplankton that are working here. And you can see we have some very small organisms down to about 5 to 10 microns. And then we have these large diatoms that form these big chains. And we can look at these pictures and identify both the health and the morphology and the species that's here. That's important to us because the diversity here has a big impact on the ecosystem. So different types of phytoplankton have a different role.
We will put out these moorings, which if you think about it, it's just like a string of instruments that is held down on the ocean floor with literally the wheel from a train, but you might have a big float that's holding everything relatively steady in the water column. So you might have a hydrophone, something that measures the speed and direction of currents. You can measure temperature, you can measure saltiness, and most recently, we've actually been putting these really cool instruments on that both measure zooplankton and fish, but also collect water samples. So they can tell us something about who's in the water when. And then there's an instrument that's called an acoustic release that when we go back to get all of our instruments back, we send it a little sound and it lets go of the anchor and everything pops up to the surface.
In our case, we were able to use a surface trawl. We had acoustics for midwater so we can look at the surface fish, the midwater fish, and we had a bottom component so that we could look at the animals on the seafloor.
The bottom trawl is a beam trawl, and so what it does is it has this big beam across the top, and that holds the net open as you send it down. And then you have roller gear along the bottom, and that between the beam and the roller gear keeps the net open as you send it down. And you're trying to catch fish and invertebrates that are hanging out right at that seafloor water interface.
Autonomous platforms are platforms that sample the ecosystem without a human on board, and they're often smaller and they can cover much greater distance than some of our research vessels can, and they can stay out for much longer at sea. So one example of an autonomous instrument is a glider, and a glider samples through the water column and it collects basic information about things like physical and chemical oceanography, phytoplankton as it swims through the water.
In 2018, we had sort of advanced technology survey. We used sail drones and acoustics. The sail drones were up there over a longer period of time. Advanced technology is important because we can't be on a ship at all times, and ships are expensive, and so we need to be able to expand our information through advanced technologies. And the sail drones allowed us to do that because they have acoustics. They are able to run the transects that we ran with our ships, and they're able to do that over a longer period of time. That doesn't mean that ships aren't important. It means that we need to be able to integrate some of this advanced technology into the research that we're doing along with ship-based observations.
You can find more information about IERPs on our website. Please visit www.nprb.org.